Today we're going to be continuing the discussion about eukaryotic gene expression, particularly the RNA polymerase II transcribed genes. And last time when we looked at this, we looked at the promoter structure, we looked at bacterial genes, and we saw how they were bound by the RNA polymerase with the sigma factors for specificity, and then we moved on to looking at eukaryotic genes, which we see are much more complicated, the promoters and the regulation. And one of the things that I pointed out was the polymerase. There's three different polymerases. We're talking about RNA polymerase II. And in addition, the polymerases are much larger. There's many subunits. And then we have what's called the construction of the transcription initiation complex, which centers around the Tata box, which is around minus 30. And the TF2D binds to that, and then TF2A, TF2B, a whole bunch of general transcription factors bind to the sequence. The RNA polymerase binds, and then we get phosphorylation of one of the C-terminal domains of the RNA polymerase, one of the subunits, and then that initiates transcription. And those are all called general transcription factors because they are bound to virtually every single RNA polymerase II transcribed gene. So there's no real differentiation between the different genes. But the fact of the matter is that the eukaryotic gene expression is highly regulated. We have about 30,000 genes in our genome, and they're not all expressed at the same time. They're expressed in all kinds of different ways, regulated in all kinds of different ways with respect to development, with respect to cell-cell interactions, with respect to treatment by different agents, hormones and growth factors, for example, environmental insults to the cells cause changes in gene expression. So we want to look at that and how the individual genes are identified and regulated in a very specific manner. So this kind of just reiterates what I was saying. That is, different cell types of a multicellular organism, they contain the same DNA. Every cell contains the same DNA, but different cells produce different proteins. That's what makes up the difference between cells. A red blood cell will express different proteins than a liver cell or a brain cell or a skin cell. And that is generally based on the specific expression of genes that encode those proteins. And of course, the cell can respond to, can change the expression of its genes in response to external signals. And we'll see later, it can be at various steps, from the DNA to RNA to protein. But one of the most crucial steps is the regulation of transcription, that is, turning the transcription on of a particular gene or turning it off. And let me just give you kind of one pretty impressive, what I think is an impressive example. So we refer to this as differential gene expression. And again, not all genes are expressed in all cell types or at all times. So we have cell type specific regulation, developmental regulation, environmental regulation, circadian regulation, which means some genes are expressed at different times during the day. We'll look at some of that in a little bit. And the vast majority of this regulation takes place at the level of transcriptional regulation, that is, turning the gene on or turning it off to be transcribed into RNA. I want to point out that not all of the regulation of the expression of a gene takes place at that level. There are what are called post-transcriptional regulatory mechanisms, which we'll talk about later. That is, you can have, for example, regulation of translation of the messenger RNA to make protein. So there's multiple levels of regulation, but the transcriptional regulation seems to be prime. So here's kind of a classic and very simple example. We have a resting cell. It's a non-dividing cell. We add, in this case, epidermal growth factor, EGF, And the epidermal growth factor is a protein which binds to the cell surface, somehow changes the communication that's taking place within the cell. And then after approximately 8 hours after the addition of the epidermal growth factor, the cells undergo cell division. So they start dividing. So the treatment with epidermal growth factor leads to cell division. And when you look at expression of genes, what it turns out, I mean, you look at what I have written here, within the first 30 minutes, approximately 2,500 genes are turned on. This is just the first 30 minutes. It's constantly changing through that 8 hours. 2,500 genes within the first 30 minutes are turned on. Often you hear of, and I have it written here, that a new genetic program is induced, that the cells change their phenotype and change their behavior because these genes are turned on. And the turning off and turning on of genes has been coined, that it's carried out through what are called genetic switches. So they're switches that turn off and on genes, and we'll look at what they look like. But I just wanted to show you a little bit about circadian regulation. So this is showing the levels of expression of what are called circadian regulated genes, that they're genes that are regulated during a 24-hour period, during a light and dark period of the day. And we're looking here, and it's analogous to what I told you about with the EGF treatment, and that is, genes can be regulated by different treatments, but here they're regulated by a time cycle. So what we're looking here is 12 different genes, all of them known to be regulated by circadian rhythm. And along the x-axis, we're looking at, it's actually a 2-day period. It goes from 0 to 24 hours, and then it starts again. So we have 2 cycles there. And then along the y-axis, for all of them, it says normalized fluorescence intensity, but actually it really represents the level of expression of that particular gene. So you can see they all have these ups and downs, and they go through these cycles. But let's just look, for example, at the one on the bottom right, which is the gene that encodes a protein called GPRK7. And you can see pretty clearly that early in the morning there's very little expression. By midday, around 10 or 12 hours, it's peaked in its expression, and then it goes back down. By the end of the day, 24 hours, it's down, and then it starts coming back up again and repeats another cycle. And this is all regulated at the level of transcription. These are called clock genes. And so people are working very hard to try and understand what is the basis of their regulation. What are the factors that actually lead to this differential gene expression? So I want to give you an example of one relatively simple genetic switch. It's actually kind of elegant, but it's simple. And this, again, we're going to go back to E. coli. And we're going to look at the E. coli tryptophan operon. So let me describe what's on here. So this is an example of a genetic switch, control of the tryptophan operon in E. coli. Okay, so first of all, what's an operon? An operon, you only find these in bacteria. An operon is a region of the genome that contains the coding sequences for multiple genes that are within a particular biochemical pathway. And they're all controlled by one promoter. So you see the promoter, and then you see what they call gene E, D, C, B, and A. Those are all genes that are required for the biosynthesis of the amino acid tryptophan. So rather than having all these genes split up on different regions of the chromosome and being controlled by different promoters, it seems to be kind of advantageous to have all these genes, which are part of that pathway to synthesize the amino acid, under one control mechanism. So if you need to synthesize tryptophan, this thing gets turned on. It makes a long messenger RNA molecule, which is shown in blue. And then that messenger RNA molecule can then be translated into the various proteins, when that shows in green, the series of enzymes which are required for tryptophan synthesis. So that's the tryptophan operon. It's regulated, in a very specific manner. So the observation is that when there's low tryptophan in the cell, so the cell is deficient in tryptophan, it means that it needs to synthesize tryptophan. What happens is the transcription of this operon is turned on in order to make more tryptophan. If the tryptophan level is sufficiently high in the cell, doesn't need to make any more tryptophan, then the operon is turned off. There's no RNA made, no enzymes, and so it maintains that balance that way. And that occurs at the level, this regulation occurs at the level of transcription, and it's through the promoter. So let's look a little bit at the promoter. Just let me point out some things here. So if you look at the promoter there, there's a sequence which is called an operator sequence. So this is between the -10 and -35 region that we saw was required for the binding of the RNA polymerase with the sigma factor 32. So this lies between that. And let's kind of blow that up on the next slide, and I'll explain how this thing works, how it gets turned on and off in that regulated manner. So here up again at the top is the promoter sequence, which has been magnified. So you can see the start site of transcription, +1. You can see the two classic promoter sequences illustrated at -10 and -35. And then the operator sequence, that's what is called a very specific DNA sequence, which is only present in this tryptophan operon. This operator sequence lies between the -10 and the -35. Now what happens is that there's a specific protein called a repressor, TRP repressor, that will bind to the operator to turn off transcription. Basically, the binding to the operator prevents the RNA polymerase from binding. So if we look at what happens under low tryptophan, which is on the left, under low tryptophan, the repressor is basically inactive, and I'll tell you in a second how it gets activated. Doesn't bind to the DNA, so in that case, the RNA polymerase can bind to the promoter and then initiate transcription. You get the RNA, you get the enzymes. In this case, the operon is on. But what happens when the tryptophan is high and the operon is turned off? How does that happen? What happens is that the amino acid tryptophan binds to this tryptophan repressor. It binds to the tryptophan repressor, causes an allosteric conformational change in the structure of the repressor such that it now binds to the operator sequence. And when it binds to that operator sequence, it physically prevents the binding of the RNA polymerase, so it turns the operon off in that manner. So here we have an on-off switch. It's on when the repressor is not bound, and it's off when the repressor is bound, and the repressor is regulated by tryptophan. Now, I personally find it incredibly amazing that the end product of the pathway, which is the amino acid tryptophan, binds to the repressor, or binds to a protein that represses the transcription of its own synthesis. This is referred to as end product repression or negative regulation. But what it's doing, in a sense, is it's measuring the level of tryptophan in the cell. If the tryptophan is high, it binds, turns it off. If it's low, it comes off, it gets turned on. So it's constantly monitoring the level of tryptophan. But the the evolutionary mechanism, in a sense, that would allow the binding of the end product to this repressor, which then binds to its own promoter, I find pretty miraculous, pretty impressive. So that's end product repression or negative regulation. Now, most eukaryotic, this is in E. coli, remember, most eukaryotic genes are turned on by activation. There is repression that occurs, but in most cases, the majority of cases, it occurs not by repression like we have here, but by binding to factors that activate transcription, which would be transcriptional activation. So let's start looking at that. So here's just an illustration that most eukaryotic genes are switched on by activator proteins. So here we have showing the RNA polymerase, which of course is simplified here because we know that it's just not the RNA polymerase. It would also require all the general transcription factors that would be bound there. But this is simplified. So you have the RNA polymerase, and what it's showing is that there's an activator protein that then binds to a site near the polymerase or near the initiation complex and then stimulates its activity. So I want to spend some time looking at these activator proteins and the turning on of transcription that occurs in eukaryotic genes. And the first example that I want to give you is of positive regulation, is positive regulation by steroid hormones. Estrogen, progesterone, testosterone, these are all steroid hormones, and they have a very specific way of turning on genes. So we're talking about positive regulation, which is common in eukaryotic cells. And the example, again, I'm going to give you is that of activation of gene expression by these steroid hormones. Like I mentioned, estrogen, progesterone, testosterone, they all have very similar mechanisms of action. And what's known as the way these hormones function to change the properties of cells is by inducing transcription of very specific genes. Estrogen has a very specific set of genes that turns on progesterone, testosterone. So they turn on gene expression of a very specific set. And I will tell you that this work that discovered this was done over 35 years ago. And just recently, over the last few years, this work has become incredibly important in certain types of cancer therapy, which I'll talk about in a minute or two. So here's what happens. So if you look at a cell, this is drawn out very simply. But on the very left-hand side, I have what's called a steroid receptor. This steroid receptor occurs in the cytoplasm. So there's a specific receptor for estrogen, progesterone, testosterone. Each one has its own. When the steroids get into the cell, they're actually nonpolar molecules. They can go right through the membrane. They go right through the nonpolar portion of the lipid membrane. They don't have receptors on the surface. They go right through the membrane into the cytoplasm. So the receptor for the steroids are in the cytoplasm. They're proteins. When the steroid binds to, let's say estrogen, for example, when estrogen binds to the estrogen receptor, it again causes an allosteric conformational change that changes the properties of this thing. It allows it to go through into the nucleus and then bind to DNA at a very specific site. So it becomes a DNA binding protein. And we know the precise sequence that these things bind to. So the estrogen receptor binds to a very specific sequence, and I have part of the sequence there, AG, AACA, so on, and it ends with TGTT. So the sequence is not that important, but the important thing is there's a very specific DNA sequence. It's called a steroid response element, SRE. And that steroid response element is located very near to where the general transcription factors and the RNA polymerase are bound on the DNA of the genes that will ultimately be induced by estrogen. So the estrogen receptor binds to its target DNA and then somehow activates the transcription to occur. So there are thought to be about 200 genes that are stimulated by estrogen. And every one of those 200 genes has this estrogen response element. So estrogen binds to the receptor, binds to the DNA, and then then the binding activates the polymerase to go. And it could be through some kind of contact that changes the conformation of the polymerase or changes the conformation of the TF2H, which then will phosphorylate. But in any case, it turns on these genes. And that, again, is what's shown on the right-hand side. And it shows the estrogen molecule going in, being bound by the receptor. Then that receptor binds the DNA. And then it shows all these cofactors, which are basically the general transcription factors. And then it says the gene is activated, messenger RNA gets turned on, and then the proteins are made. So that's the mechanism of how these receptors are activated to bind and activate gene expression, specific gene expression. Now it turns out that this very process, particularly with respect to estrogen, this finding and these discoveries have been incredibly important in understanding particular forms of breast cancer, particularly what are called estrogen-positive breast cancers, estrogen receptor-positive breast cancer. So if people have had or know people who have had breast cancer, sometimes you hear them referred to as ER positive or ER negative. What that refers to is the overexpression of the estrogen receptor on these tumor cells. About 70% of breast cancers are ER positive, and these cancers, the ER positive tumors, are a little bit easier to treat. And they're a little bit easier to treat because of this estrogen pathway that has been worked out. So let me just kind of elaborate a little bit on this. So estrogen, of course, enters the cell. There's lots of estrogen in breast tissue. It enters the cell, binds to the receptor, and what it does in this particular instance, in this cell type, is it causes cells to divide. So you have estrogen-induced stimulation of cell division, either in breast or in uterine cells. They're highly responsive to estrogen. Now, if cells are defective and have high levels of estrogen receptor, then they are more likely to be induced to divide, even if there's lower levels of estrogen that are present in the cell. So one of the major causes of the ER-positive breast cancers is that they are being induced to divide by the overexpression of this receptor. And so the natural inclination was, okay, can we block that? Can we block the binding of estrogen to the receptor? And that's one of the first specific gene-targeted cancer therapies that have been identified or have been developed, and that's a gene called tamoxifen. And tamoxifen blocks the binding of estrogen to the estrogen receptor, and then thereby blocks the ability of these cells to undergo this deregulated cell division. So it's kind of illustrated in the next slide. So the idea here is that in ER-positive breast cancers, estrogen is driving the unregulated cell division of these tumors, or at least it's participating in driving this cancer. And so what it shows on the left is that with an estrogen positive, ER positive breast cancer, estrogen binds to the estrogen receptor, and then it induces this uncontrolled cell division, because you have, when it says ER positive, it means that it's overexpressing the ER receptor. So it becomes very sensitive even to low levels of estrogen. And then the drug tamoxifen was developed. It was developed probably, let's see, in the 1990s. Tamoxifen was developed. It was a breakthrough in cancer therapy because it was the first targeted treatment. Tamoxifen will bind to the estrogen receptor, preventing estrogen from binding, and then the cell division is stopped. So what it says below is cell proliferation, which is controlled by estrogen, is now inhibited by tamoxifen. Now this does not work in ER-negative breast cancers. And so ER-negative breast cancers are much more difficult to control. They're not expressing the receptor, and so tamoxifen has no role in treatment, and so other treatments are identified, and usually they're more traditional chemotherapy type treatments. So what I want to, another thing I want to say about this is that it's kind of proves or demonstrates a principle that we hear all the time, but sometimes not always is evident. And that is, people do basic research. So the role of estrogen in turning gene expression on, and the role of the estrogen receptor, was identified 35, 40 years ago. I remember it coming out when I was in graduate school. Everybody was all interested in it because it was telling us, you know, oh, maybe this is kind of how genes get turned on, because we didn't know much about that at that point. This is how genes get turned on, at least with estrogen. Binding of a protein to the DNA activates specific genes. And then you hear things like from basic research that maybe one day it'll be useful for medicine. Nobody really understood at that time the significance of what they found. Maybe this will one day be useful for some treatment of some disease, or maybe it'll be useful someday, you know, for understanding some other diseases. And so, in fact, that's what turned out. So it shows the significance of understanding the details, the minutia of how these processes work. And so when you see a defect in those pathways, it may come many years later that then you may be able to develop a drug that would interfere with that specific pathway. Okay, so bottom line here really is that positive gene regulation, positive activation of transcription is very important in turning on eukaryotic genes and regulating gene expression. So in addition to the Tata box and the binding of the general transcription factors, there is the binding of additional factors. And those additional factors are called promoter-specific factors. So the promoter-specific factors are different than the general factors. The general transcription factors bind to every single RNA polymerase transcribed gene at the Tata box. And most RNA polymerase II transcribed genes, which are regulated, which is most of them, in addition to the Tata box, they have other additional regulatory DNA sequences, just like the estrogen receptor or the steroid hormone receptors. There was an additional binding site on those specific promoters. And so every gene has additional DNA sequences that bind to specific proteins. Here's an example. This is a simple one. It was also one of the first ones worked out, probably because it was simple. And that is the HSV-TK gene, herpes simplex virus, thymidine kinase gene. It doesn't really matter what the gene is, but the principle is what's important. So You see the line drawing with the arrow, which is +1 and -25. There's the Tata box. Could be -25, -30, somewhere around there. And then upstream of that, around 80 base pairs upstream at -80, there's a cluster of 3 additional DNA sequences that bind to specific proteins. One sequence, CCCGCC, is present twice. It binds to a protein called SP1. Another one, called the sequence CAAT, binds to a protein called CTP. The sequences between those sequence elements are not really crucial. The distance between them is actually also not very crucial. They can't be right close together, but the fact they need to be there. So in order for this gene to be transcribed, all of the factors need to bind around the Tata box, and these additional factors need to bind in order to get this particular gene to get turned on. So these are called promoter-specific regulatory proteins, or promoter-specific transcription factors, because they bind to specific promoters. The specific promoters have DNA sequences that are recognized by these proteins, of course, versus the general transcription factors that binds to every RNA polymerase II gene. So the promoter-specific transcription factors, there are thousands of them, literally thousands. So there's estimated then to be, through sequencing and analysis, more than 2,500 different promoter-specific transcription factors that have been identified. And they have different names. Okay, a lot of the names are just acronyms, abbreviations. So you have a protein called AP1, which binds to a site TGA. N means any nucleotide. TGA, any nucleotide TCA. AP2 binds to CCCC, AGGC. SP1 we saw a minute ago. GGG, NF1, SRF, MYC, P53, another one. All these bind, and there's 2,500 of these things. And all these bind to these specific DNA sequences. So it can get very complicated. All these different promoters of all these different genes, we have 30,000 different genes, which are expressed at different times during the cell cycle, during development, cell type specific, circadian regulated, environmentally induced genes, hormone induced genes. All these different genes have different clusters of different DNA sequences that will then bind to a different assortment of these different factors. So this is what regulates differential gene expression in different cells. So some promoters can have 3 of these factor binding sites. Some promoters can have 15 of these factors that need to be bound in order for the gene to be expressed. So in order for the gene to be expressed, and it needs, let's say, 15, all those 15 proteins, the promoter-specific transcription factors, all those proteins need to be expressed in that particular cell type. And so in response to, let's say, some environmental signal or some hormonal signal, then a lot of these these proteins will become activated and then bind to the promoters that need to be expressed in response to that signal. So they bind in combinations, and they, in a sense, integrate signals in order to turn on the correct gene. So I want to give you one example of how this happens, and this is in response to environmental DNA damage. And the gene I'm going to talk about is P53. That's the gene that we've been working on for many years. P53 is referred to as a tumor suppressor gene. In response to DNA damage, it gets turned on, activates a whole series of genes which would lead to, let's say, DNA repair, or it might lead to programmed cell death, eliminating a damaged cell or repairing a damaged cell. So let's take a look at that. So here's just one example of how a cell can respond to environmental signals by altering the expression of specific genes, and that's through a protein called P53, which of course is the protein I mentioned that we work on, we've been working for a long time. P53 is a transcription factor, promoter-specific transcription factor, that binds to specific DNA sequences to turn those genes on. So here's an example of how P53 works. There's a few different pathways, but this is one of them. The cells respond to breaks in DNA. DNA can be broken by various kinds of toxins, X-rays, UV light. In any case, you get broken DNA, and the cell doesn't like broken DNA. It wants to either repair it, or it will die through programmed cell death, because the goal is to eliminate cells that are severely damaged. And so, in response to these DNA breaks, you get the activation of a protein kinase, which is called DNA PK, DNA-dependent protein kinase. This is a kinase that adds phosphates to proteins. One of those proteins is P53. It's activated by the binding of DNA, and that DNA comes from the broken piece of DNA that comes from, let's say, X-rays. So you get broken ends or broken pieces. It binds to this DNA PK. The binding of the DNA causes a change in the structure, and it activates the kinase activity, and then it transfers a phosphate from ATP onto P53. And that's what's shown in the middle. So the P53 with the phosphate is phosphorylated P53. And once phosphorylated, that protein, P53, becomes active. Active as a DNA binding transcription factor, so it binds to very specific DNA sequences called P53 response elements. Those sequences, those specific sequences, are only found upstream of genes that need to be turned on by P53 in response to DNA damage. And there's thought to be probably around 100 different genes. There may be more, but let's say 100 different genes that are turned on by P53 in response to DNA damage. And that's because the P53 gets phosphorylated, gets activated, and binds to those specific DNA elements, thus activating the basal transcription complex to transcribe. You get those genes transcribed, you get the proteins made, and then the cells undergo programmed cell death or apoptosis. So the bottom line is that there are DNA sequences upstream of all these different promoters, and the upstream sequences vary. That leads to the variable binding of these different factors, and thus leads to differential gene expression in different cell types. That's the basis. There's 2,500, all kinds of mixing and matching, and you can get an enormous complexity out of that. So, upstream promoter-specific DNA binding factors. Turns out that quite often what we see is, since there's multiple sites that need to be bound by proteins, often these multiple sites are clustered together near the gene. Okay, and they're clustered together in what are called transcriptional enhancer elements. These are elements, they're stretches of DNA that have multiple binding sites for different proteins. So they're clustered together kind of like in a cassette. So here we're defining transcriptional enhancer elements. So I just mentioned often transcription factor binding sites are clustered. They're clustered in what are called transcriptional enhancer elements. And here's an example. And there's thousands of examples like this. It's just one. This is a gene for beta globin, which has been studied quite a bit. So you can see the rectangular box with the start site, -25 or -30 box, the Tata box, but then upstream between where the Tata box is and around 150 nucleotides upstream, there's an enhancer element of around 100 base pairs. And it binds to 7 different proteins need to be bound. SP1, GATA, SP1, GATA, and at the very left-hand side, NF1. So there's a bunch of these proteins need to be bound. And interestingly, there's also a transcriptional enhancer downstream of the gene, downstream of the transcribe region, around 75 base pair element, which binds to another cluster of 5 proteins. How something downstream of the gene interacts with sequences around the Tata box, we'll talk about in a second, but there can be transcriptional enhancer elements upstream of the gene, they can be downstream of the gene, also they can be quite a distance from the gene, as we'll see in a few minutes. So in order for the globin gene to be expressed, and of course it's expressed in developing red blood cells because globin eventually gives rise to hemoglobin, all the correct factors need to be present. So there you have it. It's the promoter-specific binding factors all need to be in that cell type. And the only cell type they're all there and at the proper concentration is a red blood cell or a developing red blood cell, such that they bind, they activate transcription, and that's how you get the cell type specific expression of that gene. So with this, variety of promoter-specific transcription factor binding sites, you can see how complicated it can get, generating these different combinations and leading to differential gene expression. But the truth of the matter is, it now gets a little bit more complicated because there's more factors. And I'll talk about those in the next slide. So in many eukaryotic genes, what it says is gene activation can occur at a distance. And basically what the finding is that the transcriptional enhancer elements can be anywhere from 50,000 to 100,000 base pairs away from the Tata box. And that's kind of what's illustrated in the top here. In the light green, that represents an enhancer, I should say transcriptional enhancer, the binding site for activator proteins. There's multiple proteins that bind to the transcriptional enhancers, and they can be 50 to 100,000 base pairs away. The ones we've looked at earlier were the sites like with the estrogen receptor and the p53, and even with the TK gene. Those were all relatively close to the general transcription factors, so you can envision direct interactions. But here they're very far away, and the way they do interact is by the DNA bending. So the DNA loops around, and that's what's shown below. The DNA loops around and allows the transcriptional activator proteins then to make direct contact with the transcription initiation complex. Now you can see below, it shows the general transcription factors, the RNA polymerase, and then that purple that purple area, they're saying mediator. That's what they say in the textbook, but we're going to be more specific. Those are referred to as coactivators, or TAFs. So when we talk about TAFs, it brings us back to the TF2D, which I talked about at the very beginning when we talked about transcription and the binding to the Tata box. So I mentioned the TF2D binds first, and the TF2D contains the Tata binding protein plus an additional 10 to 15 different proteins, which are the TAFs. So the TAFs, turn out, are what interact with the transcriptional activator proteins. This is elaborated a little bit more in the next slide here. So what you see in this slide is that there's multiple promoter-specific transcription factors, and there's much looping going on. See the blue, the red, they're not giving specific ones, the orange and the yellow and the other red one. And all those interact with the general transcription complex via the TAFs, or what are called, sometimes they're called co-activators. Here they're calling it mediator, but there's dozens of proteins there. So the TAFs, or the co-activators, are what bridge, they form a bridge in a sense, through protein-protein interactions between the promoter-specific transcription factors and the general transcription factors in order to allow transcription to occur. So here's where the additional level of complexity comes in. I'll kind of try and describe that in the next slide. But there are, like I said, there's 2,500 different promoter-specific transcription factors, and then there are all these various TAFs, or co-activators. And different promoter-specific transcription factors interact with very specific TAFs. So I have a drawing, and this is illustrated on the next slide. So here we're talking about TAFs, the TATA binding protein associated factor. They bridge the TATA binding protein, or the TF2D, with other general transcription factors and the promoter-specific transcription factors in order to activate transcription. There are many TAFs, many different ones. I just have a few here. TAF1, TAF9, TAF31, TAF110, TAF250, and so on. And they interact with specific subsets of these promoter-specific transcription factors. So for example, SP1, which is one of the ones I mentioned earlier, binds to two different TAFs. It can bind to TAF130, or it can bind to TAF110. MYC is another transcription factor that was also in that original table, binds to TAF250. So there's my drawing down below, a little bit simpler, but a little bit more descriptive. So you have the looping going on. You have MYC and SP1 binding upstream, far upstream. And then you have the TATA binding protein, binding to the site, TATA box, and what's coming up there represents all these different TAFs that are bound as part of the TF2D complex. And so with MYC, it binds, in order for MYC to activate the gene, TAF250 needs to be there. In order for SP1 to activate the gene, participate in activating the gene, either the TAF110 or the TAF130 needs to be there. So you see it amplifies the level of complexity and the level of possibilities of gene regulation. So there's many different TAFs, and different combinations of TAFs can be part of the TF2D complex. So in order to get a particular gene to be expressed that requires, let's say, SP1 in order to be expressed, part of the upstream transcriptional regulatory elements, in order to get the gene expressed by SP1, the binding of SP1 to its site is not sufficient. Those cells also have to express either TAF110 or TAF130. And that's what bridges the promoter-specific factor with the general transcription complex. So there's another level of complexity that is, in addition to the binding of these transcription factors upstream, the promoter-specific transcription factors, you also have to have the proper TAFs associated with the complex. So yet another level of control of transcription. Now, as it turns out, we're beginning to understand how these promoter-specific transcription factors activate gene expression. We don't know it all, but we know some, because some of these TAFs have enzymatic activity. And one of them, for example, I'm going to give you is TAF250. TAF250 acetylates histones. It modifies histones and modifies chromatin. Now, as it turns out, I'm going to show you in the next slide, everything that I've kind of shown you so far is a little bit simplistic, a little bit misleading, because the DNA in the nucleus does not exist the way I'm showing you with the site available for binding of the Tata box, with the site available for SP1 or MYC or any of these factors to bind. The DNA sequence needs to be exposed. But when we talked about chromatin, what we saw was the DNA is not exposed. It's wrapped up in nucleosomes. Let's move on to the next slide here. So if you look at the chromatin, the DNA is wrapped up in nucleosomes, in these nucleosome cores, and the histones. And so consequently, the DNA is really not available for the binding of these particular factors. And chromatin in this state, where it's bound up in the 30 nanometer fiber, is basically repressive. Transcription cannot really occur very easily because nothing's available to be transcribed. So it's not actively being transcribed. It's repressed. And so what it says here in the title, eukaryotic transcriptional regulators, and those regulators can be the promoter-specific transcription factors and the TAFs, help initiate transcription by recruiting chromatin-modifying proteins. Some of these proteins are TAFs. The promoter-specific transcription factors don't have the activity. They bind to DNA, but then they bind to the TAFs, which probably activates them, and then these TAFs have this kind of activity. So what are some of the activities that are shown? The activities are shown are a histone acetyltransferase. Histone acetyltransferase. TAF250 is a histone acetyltransferase. It acetylates histones. I'll talk about that. Probably not this lecture. I'll leave it to the next one because I don't want to overdo it here. But we get the binding of the histone acetyltransferase. That would be a TAF. That would be bound to the complex. Or you have something else which is shown on the right-hand side, a histone chromatin remodeling complex. These are proteins that bind to DNA and then unwind them. And that would eventually lead to transcriptional activation. So condensed chromatin, 30 nanometer fiber, is very hard to transcribe. The DNA is bound up and not really available. But other proteins are brought in. Some of these enzymatic activities that are carried by the TAFs are brought in. They modify chromatin such that it gets loosened up. That would then allow additional factors to bind and get the process going. Okay, so next time we're going to talk about what these modifications do. So what is the histone acetyltransferase, and what's happening, and why is this important, and how does it activate transcription? We'll talk about that next time. I don't want to go too much more because I think I've kind of said enough for today. All right, have a good day, and we will pick up next time actually with this particular slide.